Okay, today we have the pleasure of hearing from Dr. Jenny Zhou. She is professor of neurosurgery and neurosciences. She's director of the Axon Growth and Neuronal Regeneration Laboratory here at Mount Sinai. She started her education at Brigham Young University. She did her PhD at Cornell in molecular biology. She got an MD at Stanford, then did her neurosurgery residency also at Stanford and her postdoctoral training in neurobiology at Stanford as well. She is very active clinically as well as managing her laboratory, which focuses on fundamental molecular mechanisms of neuron repair and glial responses in CNS injury. And her lab is one of the leading groups studying epigenetic regulation of axon regeneration. She's a recipient of numerous honors and awards for her work, including from the American Heart Association, Craig H. Nielsen Foundation, and the White Health Foundation Award. She has a very high amount of funding here, as you can see, numerous active grants and numerous pending grants. And her research has branched into a few areas that I was very interested to see are related to axon growth and cell agility, including both natural healing after injury, such as in the spinal cord, and including mechanisms of tumor migration. So today we're going to hear from her talking about mechanical forces and cell agility in CNS diseases and therapeutic opportunities. Jenny Zhou, we're really excited to have you speak today.
Thanks, Chris, for the generous introduction. So let me share my screen. Okay, you guys see it?
Yes.
Great. Yeah, so today I'd like to share with you a few ongoing new studies talking about this sort of new concepts or understudied areas, which is cell mechanics, and cell agility. Actually, I'm not going to talk about axon regeneration today, maybe next time, but today I'm going to talk about one area that we're looking at sort of like very unexpected. And actually, maybe I'll share with you guys, particularly residents, how we even turn our attention to this new topic and some of the new therapeutic things that we're doing in the lab. So what are we talking about, right? So what is cell mechanics agility? So when you think about a cell, this is the usual textbook depiction, right? You have a kind of rounded cell, there's the nucleus. So my lab actually studies a lot about these chromatin organization, gene regulation. But today I'm actually going to talk about the cells that are not round. Many, particularly in the CNS, all the cells have this kind of very active processes. And now with the high resolution imaging, you can see the cells, the membrane is constantly turned over. The cells are filled with these membrane-bound vesicles. These are called vesicle trafficking. So they constantly intake phagocytosis, and they spit out these vesicles filled with mRNA and many of the signaling molecules. And these are very familiar kind of diagram that we see in the CNS. You have these usual players, neurons, astrocytes and glia, microglia, oligo. And this cartoon actually shows that the cells are not round, right? So they have these long processes. And if you do these videos, you can actually see, particularly microglia and also astrocytes, these processes are not static. They're constantly mobile, and they touch each other. And we also know astrocytes also extend these end plates around the BBB. So now this is complex as it is, but if you think about this, I actually got this from a recent web imaging from Google of all the companies. Google actually did a 3D rendition of just a tiny slice of human cortex. And you can see this is just, the cells are, the processes are so complex. And so the question comes, what controls this agility of these processes? And do they endure physical damage, particularly in the setting of CNS disease, like in spinal cord injury, stroke, or I also study GBM migration. So today I'm gonna talk about astrocytes, but we also have very active projects in the lab studying microglia and also neurons, axons. So astrocytes are sort of, for neurobiologists, it's people are trying to shun initially these glia. So glia means glue, right? They're gluing these neurons together. But now we know the astrocytes are extremely active. So what's interesting is this image shows a mouse astrocyte that we study a lot in the lab, and human astrocyte that you guys probably were operating, but of course you can't see the cellular level. But human astrocytes are much bigger. So diameter-wise, it's about 10-fold bigger than the mouse astrocyte. So by volume, it's really a 1,000-fold, right? It's by cubic. So this is a GFAP staining. It's a marker for astrocyte. And you can see that it's already extremely complex. But GFAP is just a cellular, it's an intracellular protein. If you actually look at the real morphology of a cell, this is using a very more advanced intracellular dye. You can see the cells are even more complex. So, today I'm going to talk about what kind of molecular signaling that prevents these, that actually sort of governs these astrocytes to extend these processes without physical damage, and and what kind of therapeutic opportunity that we can learn from this. Okay, so speaking of astrocytes, here's a very interesting diagram at the turn of century. So the left one is by a Swedish neurobiologist who, using very primitive microscope, already looking at a human fetal cortex, he already observed that these glia cells have all different kind of morphology. And this is very descriptive, obviously. So today we're going to go into molecular mechanism to understanding how these cells know to respond to the mechanical forces outside to extend these processes, and what is the sort of the contextual cues for these cells to respond. And then this is a drawing by Icahn looking at the perivascular astrocytes, and later I'm going to touch upon BBB, the brain blood barrier. So you can see he already knew back then the astrocytes extend these long end plate, we call it, around a blood vessel. Here's these maybe red blood cell he's drawing. So we're going to talk about how does astrocytes extend mechanically, sort of govern these agility of these processes, and they also, if you compromise these, would that create an opportunity for the BBB breach so that we can deliver drugs? Okay, so today I'm going to talk about 3 vignettes. They're all related to astrocytes agility. And then I'm going to talk about a molecule that really dear to my heart, which is called plexin B1. I'm going to introduce that later. So first, I'm going to talk about so-called disease-associated astrocytes, or DAA, in Alzheimer's disease. And the second is going to talk about this signaling in injury-activated astrocytes. We call it IAA in spinal cord injury. And then lastly, I'm going to talk about BBB astrocyte at the end plate junction. And then finally, at the end, I'm going to talk about how we actually are embarking on this sort of therapeutic. We developed our own function-blocking anti-human plexin B1 nanobody so that we can hopefully be used in the clinic soon. Okay, so the first is Alzheimer's disease. So when Chris introduced, my lab doesn't study Alzheimer's disease. So why and how did I even get into this? So looking back, many years ago, I was actually just attending a conference, and I checked my email, and I saw this email from Dr. Bing Zhen. I just heard about his name. I never met him. He's a system biologist or computational biologist here at Mount Sinai. He leads a huge lab studying big data sequencing, and now like AI-assisted computational biology. So he was leading a big NIHU grant looking at so-called molecular subtypes of Alzheimer's disease. So when we think about Alzheimer, we all know they have plaques, they have memory decline, but Bing is actually looking at through big time sequencing. So he sequenced hundreds of patients, Alzheimer's patients, and also control subjects banked at the Mount Sinai Brain Bank. So he sequenced, at the time was just bulk sequencing initially at the cortex and also hippocampus. But now, of course, single cell sequencing, and now also metabolism, metabolic sequencing and all that stuff. So, and then he does these computational thing. And then he told me in the email that he found a plexin B1, which sits as a hub gene in this complicated astrocyte-specific gene network in AD. So through computation, they predicted that plexin B1 should play an extremely important role of AD disease progression and also maybe severity of the disease. But they're doing dry lab, right? So they just computer. So they're looking for a wet lab, like my lab, to understand what is the function of plexin B1 in AD. So we decided to devote some energy in the lab to look into this. So that's a multi-year project. So first, let me introduce what is plexin B1. So plexin is one of the axon guidance receptor. So I did my postdoc with Marcus Levine at Stanford. So he is the pioneer of this field. He cloned the very first axon guidance molecule called netrin. Axon guidance means during brain development, these molecules are guiding the growth cone of the axon to reach the correct target. So this is very intriguing, right? Plexin should be a neuronal gene function to guide the axon. So why is plexin B1 is somehow predicted to be in the astrocytes? So this is extremely puzzling. So plexin is a receptor. So here's a transmembrane domain. So you have to think about the structure of this molecule that we're about to study. So it has an extracellular domain and also intracellular domain. So the extracellular domain is very interesting. So this is a head-on look. So this is side view. So molecular structure says this extracellular domain has this ring structure composed about these very small domains. And this ring can bend. So it's very interesting. So in fact, plexin D1, another plexin, was shown to be in the blood vessel endothelial cell reacting to blood flow. So when Bing approached me, so I was thinking, wow, maybe plexin B1 in astrocyte somehow sits on these processes that I mentioned earlier, so that they can detect or respond to these plaques that's about to develop in the brain in AD patient. So this is like a kind of very vague hypothesis that we had. Of course, we have no experimental evidence to support this hypothesis. So we decided to first start to look into this. So first, we wanted to show plexin B1, is it even expressed in the astrocyte in AD? So first, we use this patient sample of AD patient, unaffected area versus a plaque area. So you can see plexin B1 immunohistochemistry already showing this kind of brown signal is showing this corona-like pattern of plexin B1. If you co-stain with amyloid beta, so in pink, purple, pink, you can see these are early plaques and surrounded by cells expressing plexin B1. So that's a pretty good sign. It is indeed expressed in the AD patient surrounding amyloid. And then if you look at the correlation of, this is a panel of AD patient with, and also some normal subjects. You look at the severity, they use ABC score to score the severity of AD clinical score. You can see plexin B1 expression pattern correlates with the disease severity. So these are just correlation. And then now this paper actually finally, after years, we finally published last year. And reviewer wants us to show that, and not just by immunostaining, you don't see the cell, show me that the plexin B1 is actually expressed in the astrocyte. So that actually took us a while because there's no good antibody for human plexin B1 for immunohistochemistry. So we eventually turned to something called RNA-scope, which is just a new technology to detect each dot is mRNA, plexin B1 mRNA. So you can see in red is a GFAP that I mentioned. So these are plaque surrounded by these reactive astrocytes. And in each astrocyte, you can see a lot of these mRNA of the plexin B1. So this satisfies the reviewer that indeed plexin B1 is expressed in the astrocytes. We call it periplaque astrocytes. Okay, so what is the function? So here's I really like this image. You can see also in human AD brain, you see these plaques are really extremely, this is probably more advanced AD, a lot of plaques, kind of scary. And they're surrounded by these, we call it periplaque glial net. It's kind of like a fishnet. There's trying to, so the hypothesis is that these glia cells, not just astrocyte, but also microglia, they react to these plaque, and then they're trying to clear them up. For microglia, they even phagocytose. I'm going to actually talk about that too. So, so the question is, what is plexin B1 doing then? Why do you need the plexin B1 upregulation, or is it good or bad for the AD? Right? Is it exacerbating the disease, or is it actually slowing down the disease? So, so for the wet lab, we have mouse genetics. So we have in the lab plexin B1 knockout, genetic knockout. So we crossed with a mouse AD model. So you can, this, I just kind of like, even though I know for this audience might be too much, but it's just amazing that this is a human civilization. If you think about it, how many people are generating this mouse? So this is a transgenic mouse, double transgenic, meaning that you're knocking two genes. One is a chimeric mouse human amyloid APP, or amyloid precursor protein, with this Swedish mutation. So we know that this mutation is very prominent, in particularly in familial AD patient. And also that gene alone is not enough to produce plaque in mouse. So you then introduce another mutant human presenilin gene called PS1. So that's why this mouse is called APP-PS1. It's a human version of APP with human mutation that's prone to, that's predicted to be, actually not predicted, to be shown to be in the familial AD variants. So, okay, with this mouse, then we cross with the plexin B1 knockout, just to see if you don't have plexin B1, is it good or bad for AD? So very exciting. So I'm just gonna distill all the years of work into a few slides. So here, this is just APP-PS, so wild type AD mouse. You can see at about 6 months or so, you see a lot of plaques developing, kind of looking like in human. And then the plaques are surrounded by this fishnet looking, we call it glial net. So green cells are GFAP. But the real astrocytes are even bigger. Remember that I showed you the first slides, the astrocytes are even bigger. So they respond to this plaque, trying to surround it as if it's like kind of bacterial or abscess, you know. So we see there are about 2 concentric layers of astrocytes surrounding this plaque. And this image shows IBA1, the microglia, is trying to also touch these plaques and trying to clear them, phagocytose them. So we call this whole thing about the whole footprint. So we can do a calculation on average, how big is this footprint and how many of these astrocytes and microglia are present in this kind of classic stereotypical plaque. But in the plexin B1 knockout, so if you delete plexin B1, so immediately you see that the footprint is much smaller. So that's a good sign usually. So when you have such a big footprint, you can imagine the neurons nearby cannot function, right? Because this become like pathological. So the footprint is much smaller. And we see that the number of the astrocytes are smaller, but more interestingly is that the cell spacing, so the cell, so actually at the time we were doing during the COVID time, so it's kind of like social spacing, right? Cell. So this is a cell spacing. They kind of ignore the social spacing and then they really come together and they swamp onto this kind of plaque. And that also allowed microglia to do the same. So they all kind of like touch these the plaques. And the plaques also, you can already see it's much smaller than in the control mice. So that's very interesting. So when we then we wait for 6 months, so you can see that in the knockout mice, the red dots are plaques in the cortical area. You can see that generally the number of the plaques are reduced. And if you look even closely in the control mice, you can see all these different plaques in different stage of the evolution. Some plaques are surrounded by these glial nets. Some plaques are more compact. And the compact plaques are actually thought to be good. They're kind of like, if you will, like, you know, people think that they don't look like TB, the granuloma, you know. So they're like more indolent and not causing too much neurotoxicity. So you can see they can be stained by this green COS staining. So you can all kind of different plaques. But in the knockout, the plaques number is smaller, the plaques are smaller, and they're more compact. They have a lot more of these COS positive plaques. So overall, the burden of the plaque and also the composition of the plaques are all favorable for the knockout mice. So that's very nice. And then we also then looked at the neurotoxicity. So here's just using some markers, LAMP1 and ATG98. These are the neurotoxicity markers. And you can see in the knockout, they're a lot less reduced than in the control mice. And then we did the memory, working memory. So this is called mouse maze, Barnes maze test. So here's like all these holes, and you train the mice, you put them on a platform, and then if they remember, they need to go to the target hole. So there involves a lot of training. So we showed that the knockout mice indeed perform, the lag time is much reduced. So the working memory is improved in the knockout mice. Okay, so and I'm not going to go into detail, but of course, then we studied them molecularly. We did single cell sequencing in these AD with and without plexin. So we showed, so each dot is one cell. That's how we do single cell sequencing, and you can cluster them in the 6 months cortex of the mouse brain. You can see all the cells are clustered nicely into either microglia, astrocyte, neuron, endothelial cell, all kind of cells. And then indeed, plexin B1, the gene that I just talked about, is expressed in the astrocyte cluster. And if you look at subcluster of this astrocyte subcluster, they can divide into a total of 9 subclusters. So each dot is one cell and one astrocyte. And they are segregated into different cluster by gene signature. Basically, each cluster have, they call it diversity, basically. Now, I don't know if we're allowed to use this term anymore. So diversity of the cells. And you can see the subcluster 8, we annotated as the plaque associated astrocyte because they express all these GFAP and vimentin, and plexin B1. Okay, so we showed that basically deleting plexin B1 can overall reduce the inflammatory gene signatures in these subclusters. I'm not going to go into detail. It's in the paper. So then we also then wanted to study human astrocytes. So to study human astrocyte, we basically using the new CRISPR genome editing. So we used human parental, human-induced progenitor cells. And then we just knock out the, using a guide RNA to target it at the exon 3 of the plexin B1. So we generated 2 clones, and sequencing showed that indeed now it's deleted. The plexin B1 is become mutant and they're deleted. So then we use a very complicated total of 3-week protocol. Actually, it takes 3 weeks to induce the from the progenitor cell all the way to induced astrocyte. So now we can compare induced astrocyte, either wild type or this isogenic plexin B1 mutation. So then we co-culture with human microglia just to kind of recreate that in the human, in the culture dish. So the green cells are microglia, and you can see in the wild type, so the red cells are astrocyte. When they come together, they kind of form this kind of nice architecture, and that's disrupted if you don't have plexin B1 in the knockout. Okay, so just to summarize the diagram, very easy to understand. So now, after a lot of detective works, now we can understand that when you develop plaques, the green cells or astrocytes are reacting, and they have these long processes, and they express plexin B1. And the plexin B1 function is to make sure that the cell-cell spacing are maintained, which is not good for AD. So because when the cells are so apart from each other, they cannot really effectively phagocytosis the early stage of amyloid fibril. So eventually they develop a huge plaque. But if you're deleting plexin B1, then they're very happy to come closer to each other and to touch these plaque. And that overall reduces inflammation and causes a smaller footprint of the glial net. So we actually depicted this is a cover article that we just published. So we have one of our Mount Sinai, they have an office here to draw these medical diagram. So we told them to create a glial net. And there's a fish, which is basically amyloid plaque. So we show that the plexins are important for the reactive astrocytes to capture to contain these these amyloid fish. Okay, so later I'm gonna talk about this is genetic evidence. So obviously later I'll talk about our function blocking antibody that we developed to hopefully mimicking this genetic knockout so that we can try in human to see whether we can also make this happen. Okay, so now I'm gonna tell you the second story, which is more to my title, which is called cell agility. It's also astrocyte. So this is a very unexpected finding and very exciting. So it's currently in revision. So this is talking about the membrane shedding or the physical damage to the processes. So earlier in the AD paper I just mentioned, we just observed, the cell spacing. We actually don't know really what's really happening. So Haofei, a postdoc in the lab, decided to take on this. So we decided maybe we should look at live cells. So we decided to do live cell imaging rather than just kill the cell and fix them. So Haofei did these live cell imaging. So here's the cortical astrocyte. These are actually mouse astrocyte. We're currently are doing the human astrocyte as well. So he derived a cortical astrocyte or spinal cord astrocyte, and then he just keep them alive and do live cell imaging. So he put these membrane glow, which is staying the membrane, supposed to be membrane in green, but the membrane after a while gets endocytosed. So that's why it kind of got into the cell. And then we also look at the cell mechanics by looking at the spike actin, which is labels the filamentous actin. So you can see the cytoskeleton of the cell. So when he did the imaging, he showed me this, and I'm like, oh, wow, what are these green membrane amangolo labeled fragments? I thought it was a contamination of the cells of sort. But then he showed, astrocyte had the same thing. And then we eventually did a video. We saw that these are membrane that shedded from the astrocyte. So the astrocyte, basically in the mutant astrocyte without plexin B1, they just couldn't keep these astrocytes from physical damage. So this is very interesting. So to prove that these remnants, membrane remnants, are indeed dead because they're shed. Once they shed, they no longer have the ATP energy. So to show that, we use a very classic Axon 5 staining, which stains the dead cell or dead part of the cell. So indeed, in the control, you see rarely a few kind of membrane damaged, very few. But in the knockout, all of these are basically stained with an Axon 5. Yeah, so this indicates, here's a cartoon. Basically, normally the astrocytes are pretty happy. They can extend. We actually have video showing that these processes are very dynamic. They extend, they withdraw. But if you don't have plexin, the agility of these processes are compromised, and then they would just shed. They just kind of like an octopus. They couldn't withdraw these processes. So this is extremely insightful to explain all the astrocyte function in CNS disease. So, and we decide to look further. So if it's shedding, like this diagram showing, then you would expect that the membrane is breached. So indeed, when we add dextran to the cell culture, you can see a few cells are labeled entirely by this dextran, meaning that there's a breachment of the plasma. So there's increased membrane shedding, and also the extension with the video. So I'm not showing the video, but the video shows that the extension and withdrawal of these processes are much retarded in the mutant cell. Okay, so this is the cell biology. What happened when you bring them together and take a better look? So Haofei showed that if you stimulate these astrocytes, you do slightly higher density culture. So the wild type astrocytes are behaving extremely well. They kind of align together in a very nice parallel orientation. But the plexin B1, remember I told you that they shed these membranes and then they couldn't withdraw these processes. And then the membranes are very adhesive. They become kind of clustered together. So this is would be very relevant for spinal cord injury, because I'll show you later in the spinal cord injury, the astrocytes are supposed to line up in the rostral-caudal orientation so that the axon can follow this kind of direction. If you have this kind of thing, then certainly it's not going to work. So first we decide to, in the culture dish, to see whether this is what's happening. So indeed, we did here we did a DRG. This is a dorsal root sensory neuron explant. So the red are a regenerating axon. So you can see the axons are very happy to follow along the longitudinal alignment of these astrocytes in the wild type situation. If you have a mutant astrocyte, then astrocytes are number one, not only shedding, they aggregate together and they cause confusion. So the axon, when they come out, they're all vertically or become sort of misguided. So, and indeed in the spinal cord injury, so this is what we see. So first we show the plexin B1, just like in an Alzheimer's disease. Here we call it IAA, right? Injury activated astrocyte. So using RNA scopes, a beautiful image showing GFAP. These are astrocytes inside. You see all these mRNA of the plexin B1 upregulated as compared to the uninjured area. And then if you delete plexin B1, remember the astrocytes are so confused. And indeed, instead of this kind of longitudinal ligament alignment, guiding the axon, trying a few axons that are regenerating, that's all now confused. You can see they're vertically oriented and very high density and doesn't look like at all like the control mice. And when we do so the membrane shedding also activate the immune cell. So you can see in the knockout mice, LAMP1 is an immune marker showing that a lot of inflammation going on because all these dead astrocytes processes are littered all over in the injury site, and they activate the immune cell. And you can even see in the cell culture study, bring these two cell population together, you can see all these much more increased white dots. These are all activation of the immune as compared to the wild type. So this is now we kind of very happy to in vitro and in vivo data showing that astrocytes without plexin B1 can shed, and that can activate the microglia macrophages. And then functionally, you can see the scar is much bigger. This is only 7 days after. This is a sagittal view of a spinal cord injury. You can see normally it's already big enough, but in the knockout, it's just huge. And at 35, a month later, the mice just couldn't contain this injury site. The injury resolution is much more compromised when the astrocytes are confused, basically. And then the axon regeneration is much less. And here's a BMS motor sensory function score. After spinal cord injury, wild type recover. We do a mild injury, so they do recover, but without plexin B1, they just couldn't recover. So this is a conceptual kind of study showing that in the spinal cord injury setting, the astrocyte agility of these processes are extremely important. Otherwise, they would shed and they cause unwanted the neural inflammation. Okay, so now I'm gonna, this is actually a talk about the third one, which is still just cooking. We're still just, all these data are very fresh. And this is a very exciting. So we, with these two studies, so we actually, later on, I'm gonna show you that we developed these function blocking antibody. So we're talking to Mount Sinai IP office to file for patent. And one guy there, he has a biology background, but also a lawyer, right? He was like, "Hey, Jenny, since you talk about the astrocyte, would BBB somehow changed?" He was just throwing a question, and I was like, "Wow, we never, because we don't study BBB." So I'm like, "Why not?" Yeah, because these agility of these astrocyte processes, including these M plates, are compromised without plexin. So we're talking about cell mechanics, right? Actually, I did not have time to tell you the molecularly, we also know what's happening. So intracellularly, plexin B1 basically can signal to the actin and the cytoskeleton organization. That's why that sort of explains all the phenotype I just showed you. So, but this is a really wild guess. So, Hao Fei actually did the very crucial study. So I was able to convince him to give it a try. So he did this BBB study, which is to inject through tail vein, very difficult, tail vein into a mouse IV injection of this dextran, 4 kilodalton. So it's very big, much bigger than the average drug that we give to our patients, and also conjugated with the Fixie, which is in green. So then 24 hours later, we take a look. So in the normal brain, if you inject such thing in the tail vein, the vein, there is very little breach. Actually, these are probably nothing. So there's no breachment, so BBB is intact. But in a knockout mice without plexin B1, wow, we see all these green dextran got leaked out of the blood vessel. Here, these are all blood vessels. So this is a definitive evidence showing that the plexin B1 deletion, genetic deletion, can actually cause BBB breach. So we got really excited, and currently we're actually doing in collaboration with a few labs in the Mount Sinai, looking at drug delivery. We have all kinds of fancy virus and nanoparticle to deliver, hopefully in combination with plexin B1 function blocking to cause that. So, and this is just to show an early using AAV. So AAV is an anal-associated virus. It's actually approved for gene therapy for some sort of genetic, some muscle problem, congenital mutation thing. So the AAV, so this is actually already an advanced version of AAV that's developed by some lab. Very complicated, a story of its own. Basically, it's a AAV that has an engineer-specific promoter. It's a GFAP promoter that drives this whatever gene that you want. And also at the end, there is a microRNA to cause degradation in other cells. So only microglia don't, only astrocyte don't degrade this. So neuron and many other cells wouldn't like this basically. So this is a way to target astrocyte. So we showed in the plexin B1, the control mice, injecting AAV, you can actually see pretty nice. A lot of red we use a reporter mice basically. Here's all the red cells are transduced astrocyte. But in the plexin knockout mice, you can see it's a lot more. So meaning that the BBB is indeed breached. So that facilitates a viral delivery into the brain, which is a big deal. And so we're very excited to continue on. And so currently, as I said, this just to give you a flavor, just to bring this into a publication and also a patent, we need to show that we're currently crossing a conditional knockout plexin B1 to study the astrocyte, endothelial cells, so EC endothelial cell, pericyte, astrocyte. So these different cells. Currently, the data I showed you was the genetic knockout in every cell. So we need to know, plexin B1 is function, is it in the astrocyte, endothelial cell, or pericytes? So that requires genetic evidence to delete plexin B1 only in these cells and study whether you see the same thing. And then we also have different AAV. The one I showed you was specific for astrocyte, but we also have AAV only for endothelial cells. And also maybe for pericytes. We're then gonna see whether we see the same thing. And then we'll do a more fancy transwell assay or 3D. This is in collaboration with Guoha. I think he came for a talk before. So here, this bioengineer can do these 3D BBB models. So we provided the plexin B1 knockout astrocyte and also endothelial cell. He's gonna reconstruct this in a dish to see whether we can see all kind of drug delivery. And finally, we are currently working with atomic force microscopy to directly measure the stiffness of this cell processes. So that will bring the conclusion to support our model that the BBB breaches because of the cell mechanics and agility of these processes are compromised. Okay, so now finally, this is just the last thing. So we're trying to develop a patented version to blocking these plexin. So today I only talk about plexin B1. We actually have even more elaborate study on plexin B2, and now we're also trying to do the D1. So it's really exciting. So we develop. We all heard about antibody therapy, right? We use that for immune disease and stuff. So what is nanobody? So why don't we just use a monoclonal antibody? So those antibody are usually human. We call it antibody. So apparently the camelid or llama or camel. I was actually chatting in the OR with some resident. I don't know if you guys know that they actually have different their antibody is different than the human antibody. So human antibody, this is IgG, right? You guys still remember there's a heavy chain, light chain, and Fc. That's what the antibody looks like. But in the camel, they don't have a light chain. They only have this heavy chain. And this is just evolutionary conserved in that species. Very, very interesting. So with that, so you can just generate, this is much smaller than this already, but because the way they work is just the heavy chain, you can just do this VHH, or variable heavy chain. So this only is 12 kilodalton, and whereas IgG is 150, so it's 10-fold difference. And then why do we care about this? Because BBB is still an issue, right? We couldn't deliver a lot of drugs into the brain because of the too big. So this already shrink by tenfold. And also with the plexin, that we think we can even deliver even more. And also with this much smaller nanobody, you don't have to produce in the in sheep or mouse. You can produce these nanobody in E. coli or just the mammalian cell culture. So much more effective. And you can also do all kinds of combo. So nanobody is a sort of emerging therapeutic thing. So a few years ago, we decided to work with Jian Yu and Roland. We three lab joined forces. So we developed three nanobody. One is against human plexin B1, the other is human plexin B2, and another is the LRRK2, which is actually important for Parkinson's disease. So we have we clone these and then we send it to a llama alpaca farm in Pennsylvania. It's very interesting. And then they produced the serum. They inoculate, kind of like vaccination. They inoculate and then they give us the llama serum. And then we did multiple rounds of re-vaccination. And then we finally got about 20 for each. So we have 20 our special nanobody sequence. So actually, we know the sequence of these nanobody. So then we did a lot of screen. And then actually, next time, maybe I'll show you that we have drug binding assay showing that we have reached the nanomolar concentration, which is extremely high affinity. So currently we're developing, we actually already have some very exciting data showing that indeed it functions as a function blocking nanobody in some of the assays. Now we're gonna bring together into the nanobody delivery. So one delivery is just the usual IV delivery. We're still worried about BBB not efficient enough. The other way is to just using the AAV to encode this VHH domain, about 12 kilodalton, very small peptide. And also we can also produce AAV with the FC, because FC might be important for the macrophage to react to the antibody. So then we have all kind of engineering to have a more advanced level of the advanced version of the AAV. And then we're also, just to give you a flavor why we're so busy in the lab, because to bring this into the human, we need to test that, right? So the antibodies against human, but all the mice model are mouse plexin B1 that wouldn't necessarily bind to this nanobody. So we're currently generating humanized plexin B1 mouse. So the way we do is we're taking two different approach. One is something called more mature technology called a BAC, or bacterial artificial chromosome. So instead of introducing a very small peptide, we actually, there's technology, it's very, it's already 20 some years old technology, basically using a very huge bacterial artificial chromosome with our gene of interest, which is the which is a human plexin B1. So we now going to generate mice that express human plexin B1. And the second approach we decide to do is even more sophisticated. So we don't want to replace the entire plexin B1, because what about the intracellular domain may not work? It's a human version. It may not work with the mouse signaling downstream effectors, right? So we decide to make a fusion. So the extracellular domain is human, but intracellular domain is mouse. So when we have this plexin B1, then our nanobody should be able to target the extracellular domain of the human plexin while leaving the intracellular signaling intact. So this is, we already are just injected the mice. So in a few months, we would have that. And this one, we're still just cloning right now. Okay, so the team, it's a huge effort. This is just highlighted how we did the agility study. Christian did the initial cell mechanics, and Jan did the AD model, and a huge team of collaborators and funding. Thank you. So.
Jenny, it feels like you stopped in midstream. I'm sure you could keep going for many, many more discussions, and I would love to have you come back. Are there any questions or comments?
It's a really impressive body of work. So plexin B1 is in general pro-inflammatory. it seems. And if you block it, you can decrease inflammation.
Yes, yes. In a nutshell, yes. Yeah, but it's not controlling the inflammatory genes. It's controlling cell mechanics that then led to the inflammatory reaction. Yes.
Yeah. When you get the nanobody, it'd be really interesting to try that in intracerebral hemorrhage models, where perihematomal edema is a major problem.
Yes, exactly. Right, the swelling, which I didn't talk about it. So we also found that in the swelling, the osmotic challenge, the cells swell, and then that the membrane turnover is extremely important. And so there's this new concept called, in fact, actually I use A. So you guys all should use AI. The AI is amazing. You enter a natural language question, it has all the entire human knowledge. So basically, yeah, so there's this emerging concept called, you want us to make the more youthful looking cell mechanics and membrane properties more youthful. The cells are more useful. So they actually, in fact, show that in aging brain, the aged cells don't have we all know like ROS, the redox balance is bad, right? They're oxidized stress. But that's signaling. But also, if you look at these cell mechanics and agility, they're all compromised. So the membrane don't look as useful. Kind of like our skin, right? We don't look as useful when you get old. The same is for the cell. So that's what we're like touching upon this very kind of overlooked area.
Well, that's awesome.
And that mechanism you started with, so the knockouts impede the microglia and glia, which are supposed to be helpful, right? But they're causing more damage.
Yeah, so they're exactly. So it's always a two-edged sword. They claim that these glias are important. Obviously, they are important to clear the plaques and to organize the wound, but prolonged inflammation is bad. So in the plexin, so the reason it took us a while because you can't just sequence them and look at the gene. It took us a while to understand what's really going on. So in the end, we think it's a cell spacing. So the microglia just couldn't astrocyte the microglia so plexin basically is an axon guidance molecule. It tells the cell to keep a distance from each other, not to it's repellent. It's axon repel basically. Here is a cell repel. They repelled each other to the point that they couldn't even function. They couldn't even come to eat. So the cell spacing is detrimental here. Yeah, too much plexin. So if you can block, maybe not completely abolish, just a block a little bit, you let them come in to clean up the plaque. That should be very good.
Fascinating.
Yeah, and then the BBB is even more exciting. So the holy grail right now for BBB are not cell mechanics. So because nobody knows how to, I mean, cell mechanics study, people are using these like inhibitor for the actin and microtubule. Those are not drugs, right? You can't do that. So we're very happy to find the signaling, so we can target only the signaling part, so that the consequence is the cell mechanics are compromised, rather than just destroy the cell. I kind of use the analogy: you can't destroy the car. You have to like just target it. Yeah, if you disrupt the cytoskeleton, then the car cannot move. But that's not a therapy, right? So.
Jenny, if you could scale up your activity, I mean, we've discussed this one-on-one many times, but if you could just wave a magic wand, what would you like to see to really increase the capacity of your lab to do many, many more things at the same time?
Yeah, so there were I mean, it's actually interesting to discuss. I actually talked to a few people. So we first need to get the patent because we have the sequence. We need to protect the sequence. If the sequence is if I tell you the sequence, everybody can just make their own nanobody, right? So we need to patent. And then with the patent, but then when you file, it's like a chicken egg. When you file patent, they want to know what's your implication, what kind of specifics that you think the plexin nanobody can do. So we're like literally still developing. So actually, I didn't tell you So one thing we think therapeutically very interesting is the BBB. I think why if we're really can using nanobody to breach the BBB, that can help everything. It's not just AD. Any drug delivery can be helpful. But we need to convince them. And then second thing is the AD. Of course, I need to to really show in the AD. So that's why we need to generate humanized mice just to show that my nanobody can work against human Plexin B1. That's AD, I think it's difficult, but BB is very exciting. The other thing which I didn't talk, maybe next time I can talk, is very, very interesting. So when you make the cell mechanics, when you make the cell soft, that's what we're thinking. The knockout cells are soft. They just don't have that kind of. So initially we thought it was bad, but turned out to be that when you get old, the aged cells are stiff, just like, and then when you're too stiff, and guess what happened? All the stress response that we study, the DNA repair, the redox balance, and also senescence, all these stress response are not as good. So the soft cell is better. So we actually show that like after radiation or after any kind of stress, the cells, if you can make them soft or more youthful, they are very, very resilient to the stress. So I think we already show that's anti-plexin B2 nanobody. The cells can resist radiation therapy. They repair DNA much faster. So it's the very striking phenotype. So we're very excited. So yeah, so we need to show these, and then we get a patent, and then have a company, and then attract funding from the investor, and then you can have army of people to test all of these ideas, and then bring them into a clinical trial.
Right. Yeah, I was exactly thinking, how do we you need an army. Clearly, you need a whole army. You need maybe a company to do it. I mean, we should explore every possible way to get that army for you.
Yeah.
All right. I hope that the rest of the team here is inspired. It's a far reach to go from where you start to where Jenny has gone. And she's had a few students. Jack is one of them, recent student. She's had a few students who functioned well in your lab. And she welcomes students and residents who are willing to, dig in and really work hard. It would probably be life-changing to work in a lab like that. So hope it's inspiring to some of you.
Thank you.
Thanks, everyone.
Thanks, everyone. Have a great day.